Okay, can somebody confirm that you can hear me?
Yeah, I can hear you.
Yes, we can hear you.
All right, you should be able to see the screen too. Ooh, wait a minute. Well, let me stop sharing my screen. I forgot about something. And that is, I did want to mention that I did grade one of the labs graded. I think it was lab 3. Nope, lab 2. So lab 2 is graded, and I'll try and work on lab 3 tonight. Hopefully I can finish it. And then you can go through and make corrections. And as long as you turn in your corrections by 11:59 p.m. this Saturday, you will get your score from your corrections, which will be for full credit. You may get up to 5 points. Okay. A number of people turned in corrections for lab 1, but not everyone. And it looks like a number did for lab 0 too. I will go through and regrade those when I have time. My first priority is to get, of course, lab 3 graded. All right, any questions about any of that? Let me shut the grade book down. I am extremely busy at the moment, and I'm teaching another course, and that just started. So, but I will make sure I give you guys enough time, so don't worry about that. Am I sharing my screen now? No, I'm not. I can tell. I am recording though. All right, let me share my screen. If there's no further questions about that, let's begin. What we're doing today is lab 4 on stains. You'll notice on Thursday, there is no lab. I will be here for questions and answers, but after the last student logs out, I will log out myself. So there is no lab schedule for this Thursday.
Quick question, Professor, just to double check. When we're submitting our corrections, do we just press resubmit assignment for the worksheet lab? Okay, it won't mark us late or anything?
Oh, it might, because Canvas isn't set up to have 2 turn-in dates. But as long as you do it by the until date in the lab, you will be getting it for full credit, even though Canvas may tell you you're submitting it late. And that's because I have the first date that's due and then the second date that it's due. And there's no way to do that in Canvas.
Okay.
Oh, good.
Thank you.
All right, no problem. And let me remind you guys, try not to use the, what's it called? The
comment section.
Comment section. Thank you. Try not to use the comment section. I like to put my comments in there, and then you can use that to regrade your paper. Okay. If you want to put a comment in your paper, just put it in at the top of the page or the bottom of your page that you're submitting, and I promise I will read it. Oh, I didn't save that. I thought I did. The lecture just finalized, so my computer will be under a lot less strain and pressure and be faster now. I don't need this page, but I don't want to close that down yet. So let me go home. And let's begin the lesson, lab 4, staining techniques. Let me remind you, please do not turn in the lab module. It is very long and has many pages. Like, for example, this is the lab module. And it has 15 pages, complete with pictures. Turn in the worksheet, which will be at most 2 pages long. Maybe 3 pages if there's a lot of pictures. Actually, this one's 6 pages. It's a lot longer than I thought it would be, but it has pictures. Oh yeah, this one has a lot of pictures. All right. Let's see. Go back to the module. So there is some material to read on the textbook. Read about the size and the shape and the arrangement of bacteria cells, about the bacterial cell wall, about simple stains, about differential stains. And when we're talking about differential stains, there's really only 2 you need to know, the Gram stain and the acid-fast stain. And then we'll talk about special stains, and these are special stains to stain special parts or special structures within cells. And there we'll talk about the negative staining for a capsule, about the endospore staining, and about flagella staining. The flagella being a special structure a cell can have for moving or locomotion. Each of them are just a couple of pages long, but you should read that before you begin the lab. In today's lab, we have video clips and a virtual lab. In the virtual lab, you'll be using a computer program to simulate using a microscope. The learning objectives: 1, prepare bacterial smears for staining. And let me state that when you make a bacterial smear, you're not doing the staining, okay? Many times students get that wrong. When you make a bacterial smear, you're just getting a slide ready for some other use, which is usually to stain it next, but you're not doing the staining when you're making the bacterial smear. 2, understand the principles and the procedures used when performing a simple stain, a differential stain, and a special staining technique. 3, interpret the results for a simple stain, a differential stain, and special stains. And 4, to be able to define the following terms. Okay, any question about any of that? All right, because of the extremely small size of bacteria and what they're made of, bacteria are almost transparent. Therefore, it is necessary to stain them before they can be observed under the microscope. Let me blow that up a little. A bacterial smear is a drop of sterile water placed on a clean slide. So I'm going to have this slide. We put a drop of water on it, and then we add our bacteria, usually on a loop. You pick up the bacteria on a loop, and then you mix it into the drop of water, and then you smear the drop of water all over the slide. Then you allow the water to air dry. And that's why you're using a single drop, and that is the drop of water, when it's spread all over the slide, will generally dry in something like 2 to 5 minutes, generally 3 to 5 minutes. If you use more than a drop of water, it will take more time. That isn't a problem. You just have to wait a little longer, but it will dry. And then when the water is completely dry, you then fix the bacteria to the slide by gentle heating. So you get a bacterial incinerator. My couple act like a bacterial incinerator, which isn't quite right, because I'm going to pass it over here so you guys can see it. And you actually flip it so that the cells are facing the heat. And the bacterial incinerator actually, I can't do that because I got water in there. The bacterial incinerator, actually, the heat will be coming out of a hole on the side. So you put the slide on the hole that way. But we call that heat fixing, where the bacteria cells will then be attached to the slide. That's so that they will not be washed off during the staining process which is usually done after you make the bacterial smear. Particularly if you're washing during the staining, the water washing will remove the bacteria unless you air dry and heat fix that attaches the bacteria to the slide. Any question there? All right. So after you have the bacteria smear, you then usually stain them. And there's 3 types of stain. There's a simple stain where you only use one dye to stain the bacterial cells. And I don't think I've got that crossed out on yours. I should. It says a basic dye. Usually we're using a basic dye, but it doesn't matter if it's a basic dye or an acidic dye. It's still a simple stain. You're using one stain to stain the cells, and that's a simple stain. You can only tell the shape and the cellular growth arrangement when you're using a simple stain. A differential stain uses 2 or more dyes to provide additional information about the cells. So you can get the shape and the arrangement, but you can get different information. For example, in the gram stain, you can determine what type of a cell wall you have. Is it a gram-positive cell wall or a gram-negative cell wall? And then lastly, so that's a differential stain. And then lastly, we have special stains, and they're used to dye a particular bacterial structure, such as the flagella in the bacteria. So when we're looking at cells, you have to look at what type of a cell it is, and we have 3 basic shapes. There is a cockeye shape, a round cell. There is a bacilli, and they're rods. There are spirilla, and they're sort of S-shaped. If they have multiple S-shapes, we call it a spirochete, but we'll talk about that later. It's still a spirilla as the basic shape. And then there are a few other shapes, but those are the 3 main ones. Pleomorphic, the cell can take on any shape. And then there's coccobacilli, which is in between a cockeye and a bacilli. Those are the shapes. We also can look at the cells and see the cellular growth arrangement, sometimes called the cellular arrangement. And you can have singlets or single cocci, which isn't shown here. I guess this would be a singlet. You can have diplococci, and those are 2 cocci stuck together. You can have tetrads, and those are 4 cocci stuck together. You can have 8 cells stuck together. And you can have staphylococci, which are a whole group of cocci stuck together. We'll talk about staphylococci later. It actually means this looks like a cluster of grapes. Okay, and then the last shape you can have with cocci is streptococci, where the cocci line up in a line or a string. You have the same shapes in bacilli, but you have fewer growth arrangements. You can have singlet bacilli. You can have diplobacilli. You can have streptobacilli. And that's it. You do not have tetrad. You do not have staphylobacilli. Okay. Any question about that? We'll talk a little bit more about the cellular growth arrangement later. Differential stains, turning to those, there are 2 differential stains. Like I said, the gram stain is the most important. You don't need to know when it was invented, but you should know it was invented by Dr. Hans Christian Gram, and this is a person. So whenever you use the name gram stain, you should capitalize G because this stain is named after a person. Okay. The gram stain was a stain that Dr. Graham was using to differentiate bacteria, the bacterial infection, from the patient's own cells. And microbiologists started using the gram stain, and they noticed that it could distinguish between various bacteria according to their cell wall. Some cells could retain the crystal violet of the gram stain, the first stain, and keep the purple color, and these we call gram-positive cells. And then other cells cannot retain the purple cell, which is the crystal violet, and these we call gram-negative, and they're stained by the counter stain, which is saffronin, and it is pink in color. So gram-negative cells appear pink, and gram-positive cells appear purple. So here we're looking at a gram stain, and you can see various purple cells in there, mostly rods. I think there's a cockeye right there. And then you can see a purple gram-negative, a pink gram-negative rod or bacilli. We can see gram-negative spirochetes right here and right there. And that looks like a gram-negative cockeye right there. All right, in the gram stain, you get different gram-positive and gram-negative cells, and this difference is the difference in the cell wall of the bacteria. The gram-positive bacteria is different than the gram-negative, and their cell wall is very thick and has a thick peptidoglycan layer. Whereas the gram-negative cells, they do have peptidoglycan in it, but it's much thinner, and their cell wall is thinner. So this is the gram-negative cell and has a very thick peptidoglycan layer, and the gram-negative cells do have peptidoglycan in their cell wall, but it's a much thinner layer. They also, gram-negative cells have this outer membrane, which has lipids in it, and that's missing in the gram-positive cells. Any question about any of that? So after bacteria have been stained with the crystal violet, we then use Graham's iodine. Graham's iodine in this case is a mordant. Graham's iodine chemically combines with the crystal violet, forming a larger crystal complex, meaning a crystal violet iodine complex is larger than the crystal violet alone. We then take that and decolorize the cells usually with an alcohol acetone solution, and that will remove the crystal violet iodine complex from the gram-negative cells, but it will not, if done correctly, from the gram-positive cells. We then counterstain the cells with saffronin, and saffronin is a pink stain, and that will make the gram-negative cells stain pink, but the gram-positive cells will not stain pink. They will continue to stain purple. Any question about that? All right. When you're doing the gram stain, is that what this is talking about? Yep. When you're doing the gram stain, if you don't use freshly grown cells that have grown for less than 2 days, or 2 days at most, you can see a gram variable outcome, especially for a certain species, where the cells, some of them stain purple, and some of them stain pinkish. Let me see if I can blow this up. What do you notice that's different about this pink cell here and that pink cell here and this purple cell here and that purple cell there and these two purple cells here? What do you notice that's different?
The purple cells look a little thicker.
Well, that one certainly is. I'm not sure that these are thicker than that one, but yeah, they're thicker than this one here. So sometimes they're thicker, that's true. What else do you notice?
There are different shades of pink and purple.
There certainly are different shades of pink. I'm not sure if there's different shades of purple. That one might be just a little bit out of focus. But there are different shades. What do you notice about this cell or that cell? Or where's the one that looks broken? I saw it just a minute ago. I think it's this one. See how that cell looks broken? And in a sense, that one looks broken too. Well, my point is the gram-negative cells are looking broken or ghostly. It looks like a ghost of a cell. So does that one. So does that one. My guess is that these cells had grown for many more days than 2, that this is an old culture, at least a week old, maybe even 2 weeks old. And that these cells are old and dying cells or dead cells. That's probably why that's broken, because it's dead. And then these cells are newly grown cells. These are fresh. Their membrane looks intact. The cell looks whole and complete. Whereas these cells, they don't. They look almost like ghosts of cells. If you know what I mean by a ghost, something that's partially there and partially not. Okay. And that's what happens when you use old cultures. The cells may not be alive, they might not be intact, and they won't stain the way they should. Okay. When I've stained the mycobacteria, if ever you use cells that have been growing for 2 days or less, they always appear gram-positive. Now, admittedly, I've only really grown one species of mycobacterium, but if you grow them for much longer than 2 days, like a week or 2, you will stain them and you'll see gram-negative and gram-positive. But if you had grown them correctly, meaning 2 days or less, and then done the gram stain, they'll all be gram-positive. So that's your main tip for getting good staining results with the gram stain. Avoid using old cells. Another tip is prepare a thin smear. Ideally, the bacteria smear should only be 1 cell thick. Heat fix the bacteria onto the slide only after the smear is air dried. If you heat fix before the cells have air dried, you cause the water to evaporate, and that will puncture the cells. And that may lead to inappropriate staining. Avoid overheating the cells during heat fixation. Excess heat can disrupt the cells, and then the crystal violet may leak out of the cells. Of course, use fresh staining reagents and do not over decolorize. And let me add, do not under decolorize. And those are some tips for getting good gram stains. All right, any question about the gram stain? If not, let's move on to another differential stain, the acid-fast stain. Some bacteria, mainly the mycobacteria, but also Nocardia, have a waxy substance called mycolic acid in their cell walls. These bacteria are able to resist acid alcohol decolorization, meaning in the acid-fast stain, they resist the decolorization. These cells also resist a normal stain because normal stains are water soluble. And if it doesn't take up the stain because of the mycolic acid in the cell wall, the cells will be transparent. You can't see the cells. Usually, if you do the gram stain correctly on the mycobacteria, you will get the cells staining gram positively, but they might be a little lighter than normal. And that's because crystal violet and safranin are water-soluble stains, and so they are not readily taken up by the mycobacteria. And more importantly, they resist the decolorization step. So initially, with the acid-fast stain, when you're staining with the primary stain, which is carbol fuchsin, the cell will take carbol fuchsin in and look cherry red. You then stain this, decolorize the cells with decolorizing agent, which is an acid alcohol. And that will decolorize any of the non-acid fast stains, meaning the acid fast staining negatively cells. You then apply the counter stain, which will change the cell's appearance from clear to whatever the stain color is of the counter stain. The counter stain for the acid fast stain is either blue or green, and it depends on which counter stain you use. If you use methylene blue, the cells that are negative will stain blue, and if you use brilliant green, the cells which are acid fast negative will stain green. If the cells are acid fast positive, they'll retain the carbol fuchsin and they will stain red. So in this slide preparation here, the green cells are acid fast negative cells, and the red cells are acid fast positive bacteria. All right, any question about any of that? If not, let's move on to special stains. There are 3 negative stains we're going to talk about. The first, not negative stain, there's 3 special stains we're going to talk about. The first is negative stain or negative staining, which stains the glycocalyx of cells. This is also called the capsule stain because it's a special stain that stains the capsule. The glycocalyx is firmly attached to the cell wall. We call it a capsule, but even though it is firmly attached to the cell wall, it can be washed off the cell just in the normal staining procedures. Like if you dump the stain off the slide and then wash the slide gently with water, that can remove the capsule. And so if you use the negative stain, you're just flooding the negative stain onto the slide and then leaving it there, and then looking at your slide, you can actually see the capsule. If the capsule is one that is firmly attached to the bacteria cell and you can see it under the light microscope, then we call it a capsule. If the glycocalyx is not firmly attached to the cell and you cannot see it under the light microscope, the reason why you can't see it under the light microscope is because it's mostly water. You'd have to see it under the electron microscope. And then we call it a slime layer, not a capsule. The production of a capsule on a cell can be associated with an organism's virulence or its pathogenicity, meaning that this cell is worse of a bacterial invader in the body because it has a capsule. The capsule helps protect the cell from being phagocytized by a white blood cell or another phagocytic cell. Capsids are not easily seen with a normal staining method. Like I said, it'll wash off the capsule. So therefore, we use negative staining to help see the capsule. One negative stain is an India ink. And there you just put the bacteria cells on the slide, add the India ink to it. If the capsule is present, you'll see a clear region around the cell. So the dark region in this case is the bacteria. The clear region is the capsule. And then the somewhat dark region around it is from the negative stain, staining the background of the slide. Meaning the capsule is unstained, the negative stain stains the background of the slide. And then the negative stain, if it's India ink, stains the cell lightly, stains the background darkly. So this is not India ink. It says it is, but this is not India ink. This is some other negative stain. All right, the third special stain we're going to talk about is endospore staining. Maybe we're going to talk about 4. Did I talk about? Oh, no, we only talked about the capsule. So we are going to talk about 3. We talked about capsules. Endospores are special, metabolically inactive, hardy structures that are produced by some bacteria as a defensive strategy to survive unfavorable environmental conditions. An endospore is a bacterial cell, but it's a special small cell, which is hardy, and then just can survive very harsh environmental conditions. When the environment changes to becoming favorable, the endospore will germinate and the bacteria will return to its normally metabolically active state, and we call this cell a vegetative state. So if a cell is growing, it's metabolically active, it's going through division, it is a vegetative state. If it's an endospore, the cell is essentially metabolically inactive, meaning metabolism is very greatly removed or reduced. Not totally reduced, but it is almost totally reduced. And then the cell can survive harsh environmental conditions. Most of the cells which form endospores are in the genera Bacillus or Clostridium. This table is telling you some of the pathogenic endospores which are important to humans. So endospore forming bacilli, which are either in the family of bacilli, which this is a shape, and this is a family, or they may be in the family Clostridium. These two families produce endospores. And this table is telling you their name and their clinical significance. Any question about any of that? So we use the Schaefer-Fulton stain or the Schaefer-Fulton endospore stain to stain endospores. It uses malachite green as the primary stain, which will stain the endospore and the vegetative cells. We use heat as a mordant to help the malachite green get in the endospore. The cells are then decolorized with the decolorization step. And in this case, the decolorization is just done by water. Water will remove the malachite green from the vegetative cells, but it will not remove the endospore green from the endospores. I mean, the malachite green from the endospores. We then use safranin as a counter stain to stain the cells which have been decolorized. So the vegetative cells will stain pink from the safranin, and the endospores will stain dark green from the malachite green. The endospores will retain malachite green, the vegetative cells will not. So here we have a slide showing you endospores. Let me blow this up. And then vegetative cells. The vegetative cells are the reddish or pink ones. The endospores are green in color. And there we have an endospore inside its parent vegetative cell. I mean, it's surrounded here by red and surrounded there by red. Vegetative green inside, not vegetative, endospore green inside the cell. Here we have a free endospore, and it's come out of its parent cell. So that's an endospore. Any questions about endospore staining? All right, now let's talk about flagella staining, the third type of a special stain. Flagellas are a structure on cells, particularly bacteria, but some eukaryotes, which allow the cell to have locomotion, go through locomotion. So if a bacteria cell can move and swim in the broth, it's because the cell has at least one flagellum. So if the cell moves in bacteria and swims, it has the flagella, because that's the only way the bacteria can move. With eukaryotes, they can move with flagellum, with cilia, and with pseudopods. Flagella are composed of the protein called flagellin, and if the bacteria has flagella, it's just an important feature, property of those bacteria cells. When we're talking about flagella, you can find flagella in different areas of the cell surface, and we name them for that. You don't really need to know the names, but you will need to know this part or these names when you're answering the questions, because one question will talk about the distribution of flagella on the bacteria cells. If you have only one flagellum on the bacteria, we call it monotrichous. If you have several flagella, but they're all on one side of the bacteria, we call it lophotrichous. If, on the other hand, you have several flagella on the bacteria, but they're all on one end of the bacteria or the other end, we then call it amphitrichous. And then if you have flagella growing everywhere on the cell, then we call it peritrichous. It's important when you're doing the flagella arrangement that you look for the cell in the species that has the highest number of flagella. So if there's only one or a few peritrichous, but the rest are either amphitrichous, lophotrichous, or monotrichous, we don't say it's all of the trichous. We say that the cell is peritrichous. And the main reason for that is that when the cell first puts out flagellum, it'll start as monotrichous. And then as the cell puts out more flagellum, it'll be lophotrichous. And the cell puts out more flagellum, it'll be amphitrichous. The cell puts out more flagella. I'm not sure if this is actually the case, because Peritrichus is a little more than amphitrichus. But if the cell puts out flagella all over the cell, we call it peritrichus. Any question about any of that? And it takes the cell a long time to become peritrichus. It'll start with monotrichus and lophotrichus for sure, and maybe go through an amphotrichus stage. Flagella are too seen to be seen with normal standard techniques. So you need special stains to view a flagella under the microscope. Most of the flagella stains coat the flagella with numerous layers of the stain, and then you can see the flagella staining because it's several layers thick or many layers thick. All right, here we're seeing flagella. Let me blow that up a little. Wrong way. And you can see that this one is peritrichus. That one is, I think, liphotrichus, amphitrichus. No, liphotrichus. I think it's only on one side. It looks mostly on this side. There might be one starting to grow on that side. And then right there, that one might be one flagella on the cell. So that'd be monotrichus. Any question about this? All right, if not, look over the terms. You should know the terms of this lab. And then work on the worksheet. Well, let's go past the references. I think this is where the worksheet begins, but let me check. Nope, it doesn't begin at the picture. So let's go back to this one. I'll talk about it. Oops, wrong one. You're not physically going to be performing any of these activities. You're going to be doing everything online, but you do need to understand how to prepare bacterial smears for staining. And you've got to know that you make a smear before you do the staining, and it's not part of the stain. And then know the principles and procedures used in the various staining techniques and be able to interpret the results you see for these techniques under the microscope. In procedure 1, you're going to be preparing smears for staining. So view the following video clip for preparing a bacterial smear, and then another short video clip for heat fixing. Go through the steps of procedure 1 and state what are the 4 common errors when preparing bacterial smears for a stain. Hopefully I've got that labeled clearly. Oh, there it is. So here, yeah, on your worksheet, it might not be labeled as a question, but you need to answer procedure 1, question 3. Oh, wrong one.
I have a quick question.
Uh-huh.
It's not regarding the lab, it's regarding the quiz. I believe it's based on chapter 1 and chapter 2, correct?
Let me take a look at the syllabus. I don't remember off the top of my head.
Okay,
The practical quiz, I don't think it is on chapter 2. I think it is on the lab and chapter 1. Now, quiz 1 covers chapter 1, chapter 2, and the start of chapter 2, as well as lab 0, lab 1, and lab 2. Okay, but you're not taking quiz 1. You're only going to be taking the practice quiz this Wednesday. Is that right? I take that back. The practice quiz you're taking on Wednesday is going to be quiz 1. So I'm sorry.
Okay, so quiz 1, it covers chapter 1, 2, lab 0, lab 1, and lab 2.
It only covers the start of chapter 2.
The start of chapter 2.
Mhm.
Perfect. Thank you so much. I appreciate it.
Okay. All right, then do procedure 2. That's a simple stain. View the following clip or video on a simple stain. A simple stain is when you're adding just one dye. It could be acidic or basic dye. So this is not correct. I should cross that out. And then after about 1 minute, you wash off the excess dye with water and then blot the slide dry with bibulous paper. With bibulous paper, it's thick paper. Let me get an example. And all you do is you put your slide, we'll pretend this is my slide, in the bibulous paper, and you fold the bibulous paper on top of the slide, and then you can press on it gently, okay? To remove the water, you do not, the cells are on this side, you do not move the slide against the paper, and you do not move the paper against the slide. You just blot it dry. That removes enough water so you can see the slide under the microscope, even if you're using oil. And then answer the questions for procedure 2. Describe the cellular morphology of E. coli. And this is a funny picture. I will tell you, E. coli is not a spiral. Okay. Give me the shape of E. coli, and then give me the shape of Staphylococcus aureus. It's right there. And then for B, give me what is the total magnification, and it tells you what lens we're using. So you should be able to figure out the total magnification. On procedure 3, we're looking at the gram stain. We have a video clip for you to watch on the gram stain, and we have a virtual lab on the gram stain. Let me go back to the lab module because I'm not sure, not the lab module, the worksheet, because I'm not sure that the link there goes to the new virtual lab. Come on, open. I did. There we go. It'll take a minute for the virtual lab to open, and then you just click continue to get the virtual lab to go. Let me turn this down because it'll be loud, I remember.
Hi, Lab Assistant. Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria.
So we're going to stain it to take a look at it.
All yogurt has bacteria in it since yogurt. Unfortunately, this is what plain good yogurt looks like. Now take a look at this yogurt. See how the whey and water are separating? Are those air bubbles forming near the bottom of the bottle? These may be signs of contamination. This yogurt looks suspicious. In the gram stain, bacteria that stain a pink or reddish color like this are called gram-negative, and their presence means the yogurt is contaminated. Bacteria that are used to make yogurt from milk stain purple. They look like this in a microscope and are called gram-positive. Pink bacteria are called gram-negative. All right, let's prepare to prevent bacteria from contaminating our work area. We have wiped down the table with a 70% ethanol solution. We will also light a Bunsen burner to sterilize the inoculating loops we'll use later and help maintain a sterile working environment. You're going to use a striker to light the Bunsen burner.
So you
The
take
gas
your
is
mouse-
turned on low already, so bring the striker to the Bunsen burner with the flint side facing down and squeeze the striker to create sparks.
You just bring the mouse
We need to
down.
dilute the yogurt before we put it on the microscope slide. Start by placing 1 milliliter of the diluent into a test tube using a pipette. This is an inoculation loop, a small loop of wire used to collect samples. We'll use it to place a small amount of yogurt into the test tube with the diluent. Start by sterilizing the inoculation loop in the flame of the Bunsen burner. Hold it directly in the flame until it glows red hot. After it has cooled down, use the loop to obtain. Place the yogurt sample into the previously measured diluent, PBS.
So you place the loop into the bacteria. I might have accidentally clicked there. And you just go down. Same here with this one, putting the sample in the bacteria from the yogurt into the container here.
Now, gently shake the test tube for 5 to 10 seconds to thoroughly mix the yogurt sample throughout the diluent. Use the inoculation loop to transfer a small amount of sample from the test tube to your prepared slide. Sterilize the inoculating loop again in the flame of the Bunsen burner. Hold it directly in the flame and
Until it becomes
transfer
red
a
hot.
small amount of sample from the test tube to your prepared slide. Place 2 loops full from the sample onto the slide.
So use the mice to go where the arrow is.
Tilt the slide to spread the drop out slightly, about the size of a penny. Now we'll heat fix the slide. This prevents the bacteria from being washed away when we begin the staining process.
It's not saying it here, but you have to air dry first and then you heat fix.
Place the slide in a clasp, such as a clothespin, and pass it over the flame of the Bunsen burner. Be careful not to put it directly in the flame. We don't want to burn the sample. Now we begin the staining process. After the staining is complete, we can look at these slides using a microscope. We use 4 chemicals to stain a slide. We apply them in a specific order and rinse the slide off under slow running water in between each chemical. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample. Let that sit for 60 seconds.
All right, any question about this lab?
Now rinse it under slow running water. Now use the bottle of grams iodine.
All right, I'm not going to go through it here because I'm going to go through it there. So we have a video clip on the gram stain, and then we have a virtual lab making a bacterial smear of yogurt and a virtual lab of you yourself doing the gram stain. The first thing you do would be, once you have the bacterial smear, although that's the first thing you do is you make the bacterial smear. But once you have the bacterial smear and it's air dried and you heat fixed it, you first flood the smear with crystal violet, letting the crystal violet soak for 1 minute. Then you dump that off and rinse the slide gently in distilled water. Second, you flood the smear with iodine. Remember, iodine is a mordant. Let it soak for 1 minute. And then you dump that off and then rinse the slide with distilled water. The third thing you do is you flood the smear with the decolorizer, which in this case is alcohol acetone. You do it for about 5 seconds. The best way to do it is you hold the slide at an angle and then bring your decolorizing over, and then you add the decolorizing, which will then drop down. You spread it so it'll drop down the entire slide. And once the decolorizing agent stops pulling out the purple color, you stop decolorizing, meaning you stop adding the decolorizer. Any question about any of that? Now, immediately, you have to take this slide that's been treated with the decolorizer and wash it with water. It needs to be immediate because the trace amounts of the decolorizer on the cell will continue to work if you don't wash that out of the cells with water. Any question about that part? All right. You then, when you're done with that and done with the water wash, you then add saffron into the slide and let it soak for 1 to 2 minutes. Usually with our dyes, 1 minute is enough, but if your saffronin is a little on the weak side, you let it soak for 2 minutes. You then dump that off and then rinse the slide with distilled water. And then you blot the slide dry with bibulous paper, and then you look at it under the microscope, looking to see if it's gram positive, gram negative, or what, and then what the bacteria shapes are. Any question about any of that?
I have a question.
Uh-huh.
So when you apply the primary dye, both the gram positive and the gram negative will appear purple in color.
Yeah, I was going to talk
until-
about that next.
Oh,
Right
okay.
here. You can see both the gram negative and the gram positive are staining purple after you've applied the crystal violet. Okay? After
Okay,
you
cool. Thanks.
apply the iodine, both the gram negative and the gram positive will be purple. It's just that this will be a darker purple, more intense, and a little less blue, meaning a more purple. Initially, it's more of a blue purple. The iodine makes it a little more purple. You then add the decolorizing, and after the decolorizing step, the gram positive cells will retain the crystal violet iodine, so they will remain purple. However, the gram negative cells will lose the crystal violet, so they will appear clear. You then add the saffronin, and that will go in all cells, but you'll see the gram positive cells will retain the purple color, and the gram negative cells will pick up the purple, the pink color. The gram negative cells will pick up the pink color from the saffronin. Any question about any of that? You will be asked quiz questions on what color do you expect to see from a gram positive or gram negative at one of these steps in the gram stain. So go ahead and list on the table here. We have E. coli on the left, Staphylococcus aureus on the right. We do the gram stain on them, and it asks, what's the gram reaction? Is it gram positive or gram negative for the E. coli and for the Staphylococcus aureus? And then if you can, give me the cellular morphology, the shape of the cells. Are they coccus or bacillus? For the E. coli and the Staphylococcus aureus. Any question about that? All right, here's the first questions in the worksheet, asking you to give me the color that the cells will be at different steps in the gram stain. Any question about any of that? So tell me what color Staphylococcus aureus is after the primary stain. And you do need to know, is Staphylococcus aureus gram positive or gram negative? And that you can get in the lab module. Or if you did the worksheet correct, you can actually get it in the worksheet from, for example, one place is this table. All right, then move on to procedure 4. In procedure 4, we're going to be looking at the acid-fast stain. We have a video clip for you to watch on doing the acid-fast stain. We're going to review the steps of the modified Kenyon acid-fast stain, or B. What's the B stand for? Acid-fast bacilli, maybe? I'm not sure what the B stands for. Oh, acid-fast bacteria, AFB. First, you stain with carbol fusion. That will stain all cells pink. We then do the acid alcohol wash, the decolorizing step, and that will make the acid-fast B bacteria that are acid-fast negative. They will lose the carbol fusion, but the carbol fusion will stay in the cells which are acid-fast positive. And then you counterstain with the counterstain, in this case is brilliant green, and then the acid-fast negative cells will stain green in color. The acid-fast positive cells retain the reddish color or carbol fusion. All right, we have the procedure for you to follow, and then we have some smears for you to discuss. Wherever there's an arrow, you need to answer, is this cell acid-fast positive or acid-fast negative? And then for that cell, answer it there, answer it here, and answer it there. If you want, you can write it like right here. The leftmost cell is, tell me if it's acid-fast positive or acid-fast negative. The next leftmost cell on the first picture is acid-fast positive or acid-fast negative. On the second picture, the left-hand arrow is acid-fast positive or acid-fast negative. And then the right-hand arrow is, so you don't have to do it exactly here. If you don't want to, you can just type it down here. But you need to tell me what color, what acid-fast positive or acid-fast negative for each of the 4 arrows. Any question about that? And that's the only question on procedure 4. Make sure you do that. All right, then go to procedure 5. You're going to be viewing the following video clips. There will be a video clip on endospore staining and on negative staining to detect a capsule. For slide A, it refers to A in procedure 5. For slide B, it refers to B in procedure 5. And for slide C, it goes to the step C in procedure 5. So this is the Schaefer-Fulton stain. You can look it up and determine which dye it uses as the primary stain. You got to name it, and ultimately dyes what cell structure of bacteria which are produced by some bacteria to aid in their survival. And then list some examples of pathogenic endospore producing bacteria. And some is more than one, okay? And just go to the table in the lab manual to get that answer. Let's see if I still have it here. All right, here are some endospore staining bacilli that are important to man. So you can just list 2 of them here. I think that's it for A. After examples of pathogenic endospore producing bacteria, then move on to procedure 5B. This is what kind of a stain, figure B, that coats long whip-like appendages which aid in the motility. So tell me what type of a stain it is. Then state the flagellar flagella arrangement that you're seeing in this picture, and you kind of have to blow it up to determine that. There we go. After that, list some examples of clinically significant bacteria that produce a flagella. Once again, you can find that in a table of the lab module. A table or in the text. I don't see a table for this one, so let me see if I just missed it. All right, so for the flagella, you're going to have to get it from the text unless there's a table down here. Nope, no table. So I have to get it from the text. All right, list some examples of pathogenic, whoops, I already talked about that one. List the flagella arrangement and type, and then list some examples of clinically significant bacteria that produce a flagella. For C, going to figure C, this is what kind of a stain that uses India ink or nigrosin and can be used to detect the presence of what structure, cell structure, that is located exterior to the cell wall and prevents phagocytosis. Any question about any of that? And then list some pathogenic microorganisms that produce capsules. And that one I'm pretty sure is in a table in the lab module. Don't have it. Might have to get that from the text. Let me see again. Produce capsules, and I don't think there is a table on this. Nope, that's the exercises. I want that one. Yeah, you got to get it from the text. It's right here and right here. Lastly, we have 2 case studies. You need to read the case study, go through the evidence. We have pictures here. And they did a chest X-ray on this female who came in because of respiratory issues. And they did a sputum and then stain for acid-fast bacteria. And then they did the Nielsen stain. And you need to name the bacteria species which is causing this infection, which you can actually see there in the lungs. Okay. Oftentimes you just tell me the disease, and in this case, I will allow it, but I prefer you to specifically mention what bacteria species, what scientific name of the species is causing the infection. And then do case study 2. A 64-year-old diabetic patient is admitted to the hospital with soft tissue necrosis of his foot. You can see that looks really bad. A routine culture and a gram stain is ordered. There's the gram stain. They didn't do the Schaefer-Fulton stain. And then tell me, based on the findings of what they have here, what the most probable diagnosis is. You must name the species which is causing this infection. And here you need to name exactly the species. You can't just say something like diabetic keratosis or something like that. You have to say the species which is causing the infection. And you can take that from this information here and from the table in the lab manual. Species of bacteria, is it? All right, any questions? If not, I will be here until 8 o'clock to answer questions. I might be here to 8:15 because we're only about 20 minutes away from 8 o'clock. But when the last student logs out, I will log off. So if you have questions, make sure you ask it before the last student logs off. And obviously, if you're here, you'll be able to answer it. All right, go ahead and get started on the lab. Let me stop the share and stop the recording.